This is our cell cycle documentary.
Бочка бас колбасит соло, колбасер по пояс голый.
The cell cycle is a long process
involving 5 phases: the interphase, prophase, metaphase, anaphase, and the telophase.
Колбасер по пояс голый. Бочка бас колбасит соло. Бочка бас колбасит соло. Бочка бас колбасит соло.
This is the interphase.
Бочка бас колбасит соло. Колбасер по пояс голый.
In the interphase, there are 3 phases: the G1 phase, the S phase, and the G2 phase. During the G1 phase, the cell starts to grow in size. In this period of time, the cell synthesizes mRNA and protein, which is necessary for DNA synthesis. There is a restriction phase that stops the cell and changes the course of the cell. In this period of time, the cell will either be allowed to continue on to the S phase or return to the inactive G0 phase. Near the end of the G1 phase, cyclin, a protein, which is a protein's activity, is significantly increased, and something called a G1 to S checkpoint is put in place, which determines whether the cell is ready to move on to the next phase, the S phase. This checkpoint determines whether the cell has enough protein, has no damaged DNA, and no other incomplete cell growth. The S phase is short for the synthesis phase. This is the phase in the cell cycle where DNA starts to be replicated. In order to begin DNA replication, the cell constructs proteins called pre-replication complexes, which are essential for DNA to start replicating. At the same time, histone proteins are also synthesized. Histone is needed because DNA has to be packaged into nucleosomes. This is done by practically wrapping a DNA segment around histone proteins. There are 3 additional checkpoints within the S phase that checks for DNA damage and prevents further cell development if detected. Firstly, there is the replication checkpoint, which checks for problematic replication forks. It stops further replication upon detection. Secondly, the checkpoint is called the SM checkpoint. It stops mitosis from happening before the entire genome is duplicated. Lastly, the cell goes through the intra-S phase checkpoint, which helps repair DNA double-strand breaks. The G2 phase is the third and final phase of the interphase. In this phase, there's rapid cell growth and protein synthesis happening. This prepares the cell for mitosis. The G2 phase officially ends when a sufficient amount of active cyclin proteins has been reached. It also repairs double-strand breaks in sister chromatids by using the correct and other sister chromatid as a template.
This is mitosis.
Бочка бас. Бочка бас. Бочка бас. Бочка. Колбасер, колбасер, бочка бас. Колбасер, бочка бас.
Prophase happens right after the interphase. In this period of time, there are 2 sister chromatids. There are 4 phases in prophase: the condensation of chromosomes, movement of centrosomes, formation of spindles, and lastly, the nucleoli begin to break down. This is usually observable, as there is an observable difference between the interphase and the prophase. Based on observations of an onion root cell, it can be noted that while in prophase, the onion root cell no longer has the dark spot, and instead has a high amount of chromosomes taking its place. At the same time, the centrosomes start moving far apart enough for it to be observed by a microscope. The formation of mitotic spindles is absolutely necessary. Mitotic spindles are made of long proteins called microtubules, which are responsible for breaking the twin sister chromatids into 2 different cells. At the end, the nucleoli begins to break down, meaning while ribosome production is halted, the energy originally going to the nucleoli is now directed towards the process of cell division.
Колбасер, бочка бас. Колбасер, голый, голый. Колбасер, бочка бас. Колбасер, бочка бас. Колбасер, бочка бас.
Metaphase is the second stage of mitosis, where, through the perspective of an observer, chromosomes line up across the equator of the cell. These chromosomes are carrying genetic information and will remain aligned across the spindles until daughter chromatids are split into 2 cells. Metaphase only represents 4% of cell cycle duration. The reason why the chromosomes are aligned evenly is because of an even pull from opposite microtubules. There is a cell cycle checkpoint during metaphase. The checkpoint makes sure the chromosomes are aligned perfectly. At the same time, it checks that there are no faulty linkages within the cell for it to continue on to anaphase.
Колбасер голый, голый, колбасер, бочка пас, бочка пас, бочка пас, 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 бочка пас, бочка пас, бочка пас, 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 пас.
There are two major movements within anaphase, known as anaphase A and anaphase B. The first phase, anaphase A, moves chromosomes to the opposite poles of a splitting cell, which are clearly indicated by centrosomes. The second part of anaphase B, is slightly more complicated. There are multiple actions that happen. Firstly, the microtubules extend out from the centrosomes on either end of the cell and meet at the middle of the cell. Since the microtubules are pushing against each other, the centrosomes are also pushed further apart from each other. Astral microtubules pull centrosomes closer towards the cell membrane.
Бочка пас, пас, бочка пас, пас, бочка пас, пас, бочка пас, 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 пас. Голый, голый, колбасер.
Telophase is the last stage of a cell's mitosis process in an eukaryotic cell. During this phase, the chromosomes gather together in the polar region of the cell, while a nuclear membrane forms around each pair of chromatids. A nucleoli would start to form, and chromosomes discondense back into chromatin. It's sort of like reversing the effect of the prophase.
Thank you for watching.
Бочка пас,